Classical Liberalism seminar. Today it's my pleasure to introduce Matt Ridley, who is a British author. He's a journalist and a popular science writer, best known for his works on evolution, genetics, and economics. He is also a former science editor at The Economist and a regular commentator on science and technology at the Wall Street Journal, as well as a fellow of the Royal Society of Literature. Matt has written several books, including The Rational Optimist, How Prosperity Evolves, and Genome, the autobiography of species in 23 chapters. And today he's here to talk about the origin of God. Welcome, Matt.
Thank you. Good morning, everybody. Thank you, Ivan, for that introduction. And it's great to be here at Stanford. And I'm going to try and tell you what we found out, Alina Chan and me, about how this pandemic began and why we think it's important. Alina is a viral vector engineering expert at the Broad Institute of MIT and Harvard. And she was the person who was making most sense on this topic, asking the right questions and being very persistent about it in the early months of 2020. I found her reliable for journalism that I was doing, so I eventually approached her and said, can we collaborate to do a book about this, to try and get as much information as we can? We expected to be overtaken by events, that the problem would be solved while we were writing our book. Didn't happen. We still don't know how this pandemic began. And I want to stress that we don't know the answer, but we are a lot closer, I think, than we were. And we both have moved from thinking it was unlikely to have leaked from a laboratory. We now both think it was likely that happened. And I'm going to try and take you on that journey, why we think that. Doesn't respond to the clicker. There you go. Great. So just the basics. Coronaviruses discovered in the 1960s, now known to exist in basically 4 families: alpha, beta, gamma, and delta. In the alpha and beta, there are 4 that have been affecting human beings for centuries. They cause a version of the common cold. They're not the main cause of the common cold. They cause about 1/4 of common colds, and they aren't particularly harmful. In this century, 2 more coronaviruses suddenly invaded the human species. SARS in 2002 and MERS in 2012. Both caused small but very lethal epidemics which petered out. And then in 2019, another one arrived called COVID-19. Virus is actually called SARS-CoV-2, but the disease is called COVID-19. And it's very closely related to SARS. So this group here, the SARS-like beta coronaviruses, the sarbecoviruses as they're known, are the ones we're interested in. And their natural reservoir in the wild is horseshoe bats. We now know this because of the work of the Wuhan Institute of Virology, trying to track down the origin of SARS over the last 12 years or so, 20 years or so, and they have made it fairly unambiguous that the natural reservoir of all sarbecoviruses is this genus of bats called rhinolodons. Horseshoe bats are a very diverse group of bats. There's about 100 species. They're confined to the old world. You don't see them in the Americas, thank goodness. And they are very diverse in southern China. There's a particular concentration of different species in that part of the world. So the question we have to ask is, what was the journey that the virus took from a cave where it was infecting bats to a city in the middle of China? And there's basically 4 reasonable options to consider. There are other ones, but they're pretty unreasonable. So the first is that it did what SARS did in 2002, that is to say, some farmed animal picked up the virus from a bat and then infected a person. In the case of SARS, it was palm civets, which were being farmed and being eaten and being sold in markets. And the very first cases were among food handlers and animal sellers in markets and so on. So it became pretty clear what the route of transmission was. It could, on the other hand, be that someone directly picks it up from a bat and then brings it to a city, because looking around the area where the SARS virus probably originated in Yunnan, the Wuhan scientists did find that there was a few antibodies to these kinds of viruses in rural villagers who lived in this area. So it's possible that that's a route that it happened. Or it could be that the person who picked it up directly from a bat would be one of the scientists, of whom number have gone in recent years into these caves in very much greater numbers than they used to do, to sample the bats, to bring back bats to the lab, to bring back samples from their bodies and so on. And it could be that having brought the bats or their samples back to the lab and put them under experiments, that there was then an accident in a lab and somebody got infected. Those seem to us to be the four reasonable hypotheses about what happened. The key moment really is the publication almost exactly 3 years ago today of a paper in Nature. It had actually been pre-printed on the 23rd of January 2020, so it was known about from about 23rd of January 2020, from Zheng Li's group at the Wuhan Institute of Virology, which was the first announcement of the full genome of the new virus that was infecting people and killing them in Wuhan. And it said something very interesting, as well as describing the new virus. It said that there was a very closely related virus in bats. It was called RATG13, and you can see there how closely related it is to the SARS-CoV-2 virus. And the way they announced it was as follows. They said, we then found that a short region of RNA-dependent RNA polymerase, which is one of the genes, from a bat coronavirus, which was previously detected in a horseshoe bat from Yunnan province, showed high sequence identity to the new virus. No reference, no footnote, nothing. So where was it previously detected? When was it previously detected? How was it previously detected? I immediately went online and I Googled RATG13 and said, right, where's, come on, I want to know the story. How did you find this bat, etc.? They found it in their freezer, essentially, because, of course, they collected it themselves about 7 years before. But it took us a long time to find that information out. They then said, we carried out full-length sequencing. Now, that implies they did this after noticing the similarity. We later found that they actually sequenced it in 2018. So it's slightly misleading the way this is written. I think we can agree. But how did we find out about RATG13? Well, this is where non-scientists, or at least amateur scientists, become important. Somebody called Rossana Segreto, who's an Italian molecular biologist, spotted that the sequence of RATG13, the bat virus genome, was identical, 100% identical to another one with a different name, RA4991. And so she, on a blog, said, are they the same virus? I mean, could this be true? And it emerged that yes, they were. This virus had been called RA4991 until late in 2019. They changed the name just before they published it. They didn't say that they changed the name. This threw us off. It took us till the end of March to find out that actually 4991 was the name of the virus we should be looking at to find out where they found it. And how did they find it? What happened to turn up this bat virus? Well, this is where the seeker comes in. He's a young man in Bhubaneswar in India, very good at finding ways through the internet. He got some passwords to get onto Chinese academic websites and downloaded some theses. And he found a thesis at Kunming Hospital, which described what happened in 2012, which led to these expeditions that found this bat. In 2012, some men, the copper price went up in 2012. That caused people to look into reopening copper mines. There was an old copper mine in a mountainside in Yunnan, which some people started digging the guano out of. 6 of them, bat guano, I should say. This is, heaps and heaps of bat droppings at the bottom of the horizontal shaft of this mine. 6 of them got sick. They were taken to Kunming Hospital, which was 600 miles away. It wasn't very close, so this was a sort of serious medical event. 3 of them died, The other 3 eventually recovered, but after months in hospital. And every top virologist in China was involved in trying to work out what they caught, because the immediate suspicion and the final conclusion was that they had picked up another of these SARS-like viruses from the bats and the guano of the bats. But they didn't quite pin down what it was. Samples were taken to Wuhan from this mine to the Wuhan Institute of Virology for analysis. And the Wuhan Institute of Virology and 2 other groups from Beijing then mounted expeditions to this mine shaft to sample the bats in the mine shaft and see what they could find. The Wuhan team went back to that mine shaft 7 times, we now know, over 2 years. So they were very, very keen to find out what was going on in this mine shaft. And let's just look at the geography here. The Wuhan is obviously up here. And this is where the Mojiang County, where the mine shaft is, closer to Calcutta than it is to Wuhan. It's a very long way, 1,885 kilometers from Wuhan. So it's not next door, which was sometimes the impression one got from the media at these early stages. It's a long way away, but they're making these expeditions over 2 years to try and find it. And one of those expeditions in 2013, they found a virus called RA4991 that was SARS-like, but not very SARS-like, distinctly different from the original SARS. And that's the one that turned out to be similar to SARS-CoV-2 6 years later when that broke out. This is a picture of them sampling at the site. It's the only picture we have. Shows them taking significant precautions, which is interesting and useful to know. And if we lighten the picture a bit, we can actually see the entrance to the mine shaft in the background. You can see the sort of boarded up entrance and the sign which presumably says keep out. A lot of journalists tried to go to this site in 2020. They were all turned away or arrested. John Sudworth of the BBC describes it as this, like this. By now you get the idea. It's impossible to overstate just how large and coordinated the effort was. State security, plainclothes police, uniformed police, officials and local residents. When we tried to talk to anyone, they turned their backs. Just to fill in a bit more of the sort of family tree of this virus, a number of other viruses did turn up over the next few weeks and months, either found subsequently or rediscovered in databases that had already been collected. And what they showed was that the closer you were to Yunnan, the more likely you were to be closely related to this virus. So the ones from Thailand and Cambodia and Eastern China and Japan were pretty distantly related. So it looks like it's somewhere around Southwest China that this thing has probably come from. None found near Wuhan, not for want of looking. The Centers for Disease Control in Wuhan, working with the Wuhan Institute of Virology, had been sampling bats, horseshoe bats, around Wuhan on a massive scale. More than 10,000 bats had been sampled over the previous few years, looking for relatives of the SARS virus, and they had not found any. They'd found a few SARS-like ones, but none that were like this particular group, none like SARS-CoV-2. Now, as you know, suspicion immediately fell on the seafood market in Wuhan, the Huanan seafood market, which was closed on December 31st. A lot of the early cases seemed to be people who'd either worked there or shopped there. And so it was immediately thought that it was like the case of SARS in 2002. It was probably an animal for sale in that market. Just to give you the geography here, the market is on the left bank of the Yangtze River here. Right next to it is the Centers for Disease Control, where a lot of the bat collecting had gone on and collaborating with the Wuhan Institute of Virology. The Wuhan Institute of Virology was in the process of moving from their old campus to their new campus a lot further south. And they had been collaborating with Wuhan University and Mojiang Agricultural University on a lot of their virology experiments. So these are the main institutions that we're interested in. A paper came out a few months ago from Michael Warraby and colleagues in the University of Arizona saying, look, it did come from the market. And here's how we know. Because the early cases are clustered around the market, even those that didn't actually go to the market. And that's pretty interesting. And he's right, it is interesting, but it's not at all conclusive, partly because there's around 100 early cases missing from this data. Around 100 cases that the Chinese authorities haven't given us the location from. So that there's, this is only about 2/3 complete as a data set. His geospatial mapping was also heavily criticized, but also there's a real significant problem here, which is called ascertainment bias. Namely, that in the first few weeks of the pandemic, the Chinese authorities said, this has started in the market. So any doctor or hospital that sees a case of pneumonia, ask them if they were at the market or if they live near the market. And if they are, check if it's COVID. So the definition of COVID was that you'd be near that market. So that's a slight problem. It's a sort of circular argument. But nonetheless, it's pretty probable that the market was an amplifying place. It was a place where the pandemic went from just a few cases to many more cases. But whether it actually started there is more difficult to say. So what about the animals for sale in the market? Well, we found out later there'd been actually a survey of wildlife trade going on in that market before, and so we know roughly how many animals of different species were being sold live in the market, a lot of them illegally. There's quite a lot of animals there that could be susceptible to catching this kind of virus. It's mostly a seafood market. It's not like the big markets in southern China where there's a huge amount of wildlife trade. It's much less common in this part of China for wildlife trade, but nonetheless, there were animals there. Notice though, no bats and no pangolins on sale. We'll come back to the significance of that in a minute. The Chinese authorities tested the animals they found in the market and came up with no infections. They could find no infected animals or samples in the market. They could find the virus on countertops, doorknobs, in the sewage, and so on. So the virus was circulating, but it was the human version of the virus. They couldn't find a 99% cousin in an animal, which was what they were expecting to find. And by 2021, they had sampled 80,000 wildlife samples from all across China and still found no animal, apart from bats, carrying a similar virus. So the search for a SARS-like chain of infections came up blank. There is to this day zero direct evidence of an intermediate animal being the chain of infection. Which leads us to talk about the possibility that it might have been something to do with the work they were doing in the laboratory. Now, people often say, well, we've had pandemics before, they've never started with labs. A, that's not quite true, and B, we live in an era when suddenly there's an awful lot more virology going on in labs than there was before. And besides, there have been a lot of cases of people catching viruses in labs. In 1967, the Marburg virus was first discovered in Germany when it started killing people who were handling monkeys in laboratories. In 1977, there was an outbreak of flu, which went worldwide, which turned out to be an extinct version of flu that was not circulating in the wild, but was kept in laboratories. And it was eventually traced back to an experimental vaccine being used in China, in northern China. In 1978, a woman died of smallpox infection in Birmingham as a result of a ventilation issue in what was thought to be a secure lab. In 1979, really crucial event, there was 65 people killed by anthrax in Sverdlovsk as a result of a leak from a biowarfare plant run by the Soviet Union. The interesting thing about this one is that they denied it for 7 or 8 years. And only when the Soviet Union collapsed and whistleblowers came forward did we find out the actual truth. Until then, the official version had been this was just food poisoning. In 2003 in Kunming, laboratory mice infected researchers with a hantavirus. and the interesting thing about this one is that the person who investigated this and pinned down the cause as a lab problem was Mojiang, the head of the Wuhan Institute of Virology lab that is at the center of this story. In 2003 and 4, after the SARS epidemic was over and there was no SARS circulating in the wild, researchers caught SARS on at least 6 occasions. Once in Singapore, once in Taiwan, and at least 4 times in Beijing. The interesting thing here is that on 5 of those 6 occasions, we still don't know how they got it. There was no dropped flask or punctured glove. They simply were working with SARS in the lab and then they picked it up somehow, despite the precautions they were taking. In 2007, foot and mouth virus broke out on a farm in Surrey in England, 13 miles from the world's leading reference laboratory for foot and mouth viruses. That turned out not to be a coincidence. A contractor had been mending a broken pipe at the lab and he'd gone straight to the farm. There's a similarity there with what we're talking about here. In this case, a sarbecovirus epidemic has begun 8 miles from the world's leading laboratory for researching sarbecoviruses. Wuhan is not just some lab, it's the only place in the world that's been doing this intensive research on sarbecoviruses to the same extent, with the exception of University of North Carolina, Chapel Hill, which I think we can agree is quite a long way from Wuhan. In 2018, the US Embassy, which had been involved in some contacts with the Wuhan Institute of Virology because of advice on how to set up their new biosafety level 4 lab, expressed significant concerns about the biosafety procedures and processes and training at the Wuhan Institute of Virology. And in 2019, just before the pandemic, there was actually an outbreak of brucellosis across China, which was traced to a laboratory leak in itself. And finally, of course, SARS-CoV-2 infects people in laboratories. We think probably quite often. It's very hard to tell, of course, because it's circulating. You might have got it in, outside the lab, or you might have got it inside the lab. But we know of one case in Taiwan where it certainly was that the researcher caught it from an experiment that went wrong. So it's not at all implausible that this could have begun with a lab leak. Now, February the 1st, 2020, 3 years ago tomorrow, there was a meeting online between a dozen scientists organized by Jeremy Farrar, the head of the Wellcome Trust in the UK, but involving Anthony Fauci here in the US and a number of other people. And that meeting was to discuss concerns that a number of virologists had that it looked like this virus was engineered, not just leaked, but engineered in a lab. And we found out about that meeting. We tried to find out what they were saying to each other before and after, and we hit a pretty blank wall, as you can see. A great deal of redaction of the emails in the Freedom of Information requests. Eventually, though, we have been able to get unredacted versions of these emails through other methods. Just to give you a taste of the kind of thing that's going on here, 2 people called Christian Anderson and Eddie Holmes both raised the possibility that it looked like this virus was more than likely not engineered. And this meeting was convened to discuss their concerns. And so here's what Tony Fauci says to Jeremy Farrar late on Friday, the 31st of January, but early on Saturday, the 1st of February. I don't think he was writing at midnight. I think that's Farrar's email time. He says, I just got off the phone with Christian Anderson and he related to me his concern about the furin site mutation in the spike protein of the currently circulating virus. I told him that as soon as possible, he and Eddie Holmes should get a group of evolutionary biologists together to examine carefully the data to determine if his concerns are validated. He should do this very quickly, and everyone agrees with this concern. They should report it to the appropriate authorities. I'd imagine that in the USA, this would be FBI, and in the UK, it'd be MI5. I think it should actually be MI6, but never mind. So this shows significant concern. What was it that they were worried about? Furin cleavage site, FCS. That is every sarbecovirus we've ever found to this day, except one, does not have this little insertion of genetic material here, which is beautifully designed to attract the attention of a human enzyme called furin, which primes the virus to be way more infectious. Without that thing, we would not be having a pandemic. Now, a number of scientists over the past 10 years, including in Wuhan, had been putting furin cleavage sites deliberately into viruses to make them easier to grow in the lab and to do experiments on them. So when they saw that, and they said, I've seen this in a SARS-like coronavirus before, that was what alarmed them. And here's Jeremy Farrar the day after the meeting, saying to Francis Collins what he thinks might have happened. A likely explanation could be something as simple as passage of SARS-like coronaviruses in tissue culture on human cell lines under biosafety level 2 for an extended period, accidentally creating a virus that would be primed for rapid transmission between humans via the gain of a furin site from tissue culture and adaptation to human ACE2 receptors via repeated passage. So even after the meeting, they're still worrying about this. But in the next few days, they draft a paper to be published, which says that we can rule out a lab leak altogether. And the lead authors on the paper are Christian Andersen and Eddie Holmes, the very people who were worried. So what changed their minds? That's the question we want to ask. And why didn't they say in that paper, we are seriously concerned, but we think it's okay? Why did they say, no, we can rule it out? Well, there's one clue as to what changed their minds, and that is the pangolins. Jeremy Farrar writes in an email on Friday, February the 7th, a few days later, reports coming out overnight that the Chinese group have pangolin viruses that are 99% similar. This would be a crucially important finding, and if true, could be the missing link and explain the evolutionary link. Francis Collins replies that would be very interesting. And he asks the right question, does it have the furin cleavage site in it? Back to the answer to that in a minute. So what's the pangolin story? Well, on February the 7th, the university in southern China announced that they'd found a 99% similar virus in pangolin. It turned out to be wrong. It was 90%. That makes a huge difference. It's way too distantly related to be the ancestor of the pandemic. But we'll leave that on one side for the moment. What's the story behind this? There's a significant and growing illegal trade in pangolins captured in Africa and Southeast Asia through Vietnam and into China. Significant efforts by the Chinese to interdict this trade in recent years. And in March 2019, they intercepted 150 smuggled pangolins, 50 of which were already dead, and the rest of which died pretty quickly. They did heroic efforts to save some of them, and they couldn't keep them alive. You can't keep pangolins in captivity. They're anteaters, they need fresh ants to eat. They're solitary animals, they quickly pick up infections in captivity and so on. Of the ones that died, some of them very obviously had respiratory diseases by the time they died. And this Southern Agricultural University in China had looked at what viruses were in their lungs. And in 2 of the 11 they looked at, there were very small signs of a coronavirus. This was then sequenced, and this was sequenced after the pandemic began, and the sequence turned out to be very similar to SARS-CoV-2. But as I say, only 90%, not 99%, as they'd originally announced in the press release. Does it have a furin cleavage site? No, it doesn't. There's the pangolin virus sequence, the protein sequence. It's missing that crucial arginine-rich RRAR sequence. It's the arginines that attract furin to that site, which is exactly the right place to split the protein in 2. And underlying that sequence in the human version is 12 letters of genetic code that have been added to the virus. Now, interestingly, for the two arginines, it uses the code CGG. That's an unusual codon to use in viruses. Viruses almost never use CGG for arginine, but humans do a lot. If you want to set it up to be effective in human cells, CGG is a good one to choose. That's a pretty esoteric point. Through the work of my co-author, Alina Chan, the 4 papers that appeared about the pangolins then turned out to be extraordinarily misleading and messy. Some of the data was repeated in different papers under different labels. That's positively fraudulent. And there were many other problems, and they eventually had to publish corrections to the papers. So it looks like the pangolin story was sort of thrown on the table as a way of distracting us from other things. That may be a bit unfair to say, but it's a possibility that one can't rule out. The Christian Andersen et al. Paper came out in March saying that we can rule out a lab origin, and citing the pangolins as one of the reasons, despite the fact that it didn't have a furine cleavage site and wasn't very closely related. This paper fooled me. I was interested in the topic at the time. People were asking me, what do I think? And I was saying, no, it's a paper that's come out that's ruled it out. You can rule out a lab origin. It took me a while to get my head around the arguments in the paper and realize that actually their arguments weren't nearly as strong as they were. And I felt very betrayed when I later discovered that they'd been having meetings in which they thought actually it was a strong possibility a few weeks before. So what were they doing in that lab? Well, the person who knows best in the West is Peter Daszak. He's the head of the EcoHealth Alliance, an organization that channeled US taxpayers' funds to the Wuhan Institute of Virology and some other research collaborations elsewhere in the world, but this was their biggest partner. And in an interview in 2019, December 2019, while the pandemic was starting, although before he or anyone else knew about it, he gave this interview at a meeting in Singapore. Coronaviruses are pretty good. You can manipulate them in the lab pretty easily. The spike protein drives a lot about what happens. You can get the sequence, you can build the protein. We work with Ralph Barrick at UNC to do this, insert into the backbone of another virus and do some work in a lab. What he's saying there is that the technique which Ralph Barrick has invented and which he has co-published with Xizheng Li on the Wuhan Institute of Virology, and which is now being used in Wuhan routinely, is whenever you find a new SARS-like coronavirus, a new SARS-CoV virus, instead of growing it in the lab, which isn't very easy, unless you've got a really good sample, you simply sequence the spike gene, work out what the spike gene is, then manufacture a gene with that sequence, and stick it into a virus that you can grow, swapping out their spike gene. Then you infect human cells with it, see how dangerous it is in human cells, and then you infect human mice with human genes in them, see how dangerous it is in that. So these are chimeric viruses. These are viruses with the backbone of one virus and the spike gene of another, used to test the dangers of that spike gene. And the purpose of this research was to predict and prevent the next pandemic. Right. And there was a lot of criticism of it before the pandemic, saying this is like looking for a gas leak with a lighted match. You may well cause the very thing you're worried about. And it probably won't work in terms of finding the needle in the haystack of the virus that might cause the next pandemic. Peter Daszak and Xizheng Li had published a paper, had prepared a paper before the pandemic began, which was in the process of getting published, and it came out in 2020. This was quite helpful because it told us, it was a summary of all the viruses they'd found and how they were all related to each other. And it talks about 630 new coronavirus sequences that they'd found. So that enabled us and others to have a good look at what they had been finding with these searches in wild bats. And two people in particular quickly, the point is, when you publish a paper like this on genomics, you have to upload the genomes of any viruses you find into an international genomic database so they're available for people to look at, which they then did in the middle of 2020, sometime after the paper came out. And Baba the elephant, a French scientist whose name I still don't know to this day, and Francisco Ribera, a Spaniard whose name is Francisco Ribera, between them, then found out something rather interesting. They found that by going through the sequence, Daszak and Zhizhengli had not drawn attention to the fact that there were 8 viruses in that sample that were very similar to RATG13 and SARS-CoV-2. Well, it would have been nice to have told us that, wouldn't you? I mean, you're publishing this in June 2020, long after the pandemic begins. There they are. They're called the 7896 group. And what Francisco realized was that their serial numbers indicated that they'd been collected from that same mine shaft. Eventually, Zhizhengli was asked to update her paper from Nature, and she printed a correction saying, yes, there are 8 other viruses from that mine shaft that we collected among the 1,322 that we collected. By then, Francisco had worked out that they collected 1,320, so he was only out by 2, but he was piecing it together from his own work. So this is the kind of sleuthing, the kind of forensic work that's been necessary because we can't get straight answers out of the Chinese scientists if we ask them. I've never had a reply to my many emails to Zhizhengli, for example. The Zika then found another thesis, and he does, and it's by somebody called Ping Yu, who's in Zhizhengli's lab, and it studies, it looks at some of these viruses, and it includes this group of 8 plus 4991, as it was still being called in 2019, and characterizes them. But interestingly, when Ping Yu published this as a paper, she omitted all mention of these 9 viruses. Now, this was before the pandemic. This was early in 2019. So there's a suspicion here that there was a degree of secrecy about this particular group of viruses before the pandemic. Now, that I don't think that means that it was part of a People's Liberation Army project to produce a bioweapon or anything like that. I think it's more likely that what they identified this group of viruses as ones they wanted to do a particularly interesting experiments on that might lead to a significant Nature paper, and they wanted to keep them under wraps until they were ready to publish more about it, something like that. Then in 2021, a new piece of information dropped, which was that French scientists working with other scientists in Laos had found another virus in a bat in Laos, in a wild bat, a Rhinolophus horseshoe bat, and it's slightly more similar to SARS-CoV-2 even than RATG13. So that's interesting, and it seems to suggest that we're on a wild goose chase chasing down RATG13 and the mine shaft, because this one's in a different country. This is in Laos. Interestingly, it's less similar throughout most of its genome, but it's more similar in the spike gene. So, you know, you can argue whether or not it's more similar, but overall it's 96.8% the same as SARS-CoV-2 instead of 96.2%. So 0.6% of the difference. But Alina and I quickly pointed out that the fact that it was in Laos didn't necessarily exonerate the lab, because we've seen reports of the EcoHealth Alliance collecting viruses in Laos and sending them to Wuhan. I published something on this. The EcoHealth Alliance sprang into life on Twitter, and said the emails cited by Matt Ridley do not show, as he claims, that we were sampling bats in Laos and sending them the results to Wuhan. So I replied, well, please explain then this entry in a database showing Laos, where it was collected, Institute of Virology, where it's stored, Peter Daszak, who collected it. Looks to me like pretty good evidence. I can't get a reply. I've repeated this request regularly since and have had no reply. So what do we know about the viruses they had in the Wuhan Institute? Because of that paper summarizing their work up to 2016, we've got we think we know most of the viruses they had up to the end of 2015. We know almost nothing about the viruses they collected after that. And it would seem obvious if they want to exonerate themselves to come forward and say, these are the viruses we collected after that, none of them look like SARS-CoV-2, what are you on about? And they have a database. It's called the Batten Rodent-Borne Viral Pathogen Database. It has 22,000 entries. It's got details about all the viruses they've collected over the years. Some of it was openly available, but some of it was behind a password protections section. And it went offline altogether. It was taken down on the 12th of September 2019, 3 months before the pandemic began, 2 in the morning, we later found. Why? The one thing they could do to exonerate the lab would be to open up this database and say, look, here's the viruses we've got. They don't explain the pandemic. They won't do that. And when we ask why not, they say because someone might hack it. If you've published it, there's no point hacking it. Do you see what I mean? So this makes no sense. What we do know is what the results of some of those chimeric virus experiments were, where they juiced up viruses by swapping new spike genes into them. And they show that in some cases, they produced a 10,000-fold increase in the infectivity of viruses in human cells. In other cases, they produced a 2-fold increase in the lethality of viruses in mice. So these were significant gain of function experiments. And it's worth just going back to their paper published in February 2020 and looking at the dog that did not bark in the night. They said a lot about the spike gene of the new virus, SARS-CoV-2, when they published this paper. They drew attention to various parts of it, including the red boxes on this chart, but they didn't mention the furin cleavage site. Now, as Alina puts it, that's like describing a unicorn, not mentioning the horse, the horn, going on about its hooves and its mane and things you don't mention. This is the most striking thing about this virus, as Christian Andersen and Eddie Holmes immediately spotted in the days after they published this. And worse than that, when they published this chart, they said, this is the sequence of the first half of the spike protein, S1. See, this is of the S1 protein. And yet it stops just short of the end there. There's plenty of space for another few spaces. It's 10 spaces short of the end of the S1 section of the spike gene. So there's exactly enough room to finish that sequence, but they've left it out. They've truncated it. Why would they do that? I mean, this is the sequence. The top one is SARS-CoV-2. All the others are these closely related ones, including the Laotian one, Bernal, 2052. And they truncated their chart there. The actual end of the S1 gene is there. There's plenty of room on that chart to put it in, but if they'd done so, it would have revealed that it has a furin cleavage. And that they didn't particularly want to draw attention to, it seems. By far the most worrying document that we came across was leaked in September 2021. It's called Project DEFUSE. It's a grant application from Peter Daszak of the Signifio Health Alliance to DARPA, asking for $14 million to do a lot of pretty wacky experiments in this field with the Wuhan Institute of Virology, including spraying antivirals into caves and weird things like that. But it included a plan to put a furin cleavage site into a novel SARS-like coronavirus. Here's the key section. We want to test them in human airway epithelial cells and in transgenic mice. any viruses that we find, that is. We will look for furin cleavage sites. If we don't find them, we will introduce appropriate human-specific cleavage sites and then evaluate their growth potential in monkey cells and human cleavage cells. So that really is a proposal to put a horn on a horse to make a unicorn. Now, it wasn't funded. DARPA turned them down. But this was in 2018, this application. And of course, the Wuhan Institute of Virology, most of its funding comes from the Chinese Academy of Sciences. So it's easily possible that these experiments went ahead anyway, or might have already been happening by now, because as you know, quite often you apply for a grant for something you've already started to do. So I'm going to stop there, but let me end with just this question. Why does it matter? I'm constantly told by people that I should let sleeping dogs lie, not rock the boat, look out for international harmony, don't provoke the Chinese. There's nothing we can do about it anyway. There's been a pandemic, we can't help it. Well, I've got three answers to that. One is that if we don't know how this one started, we don't know how to stop the next one. And that's really important. Two is that bad actors are watching this, rogue states, terrorists, and saying, wow, we could cause a lot of damage with a few virology experiments. And best of all, they wouldn't even come looking for us. They'd just wave their hands and say, we don't know how it began. And thirdly, well, between 10 and 20 million people are dead, and we owe it to them to find out why. Thank you very much.
Okay. Unrelated questions. So Jeff has a question, then Jonathan Berg.
So if I could expand on something you just sort of touched on a little bit. I'm an economic theorist, and most economic theory today involves models where there is asymmetric information. Some people know things that others don't, and involves making inferences from actions people take. I don't know how one can look at the actions of the Chinese government, shutting down the Wuhan Institute of Virology, disappearing many scientists who work there, scrubbing research, refusing to allow any real investigation. I don't know what story one could possibly tell of that without leading to an inference that the Chinese government thinks that the virus was related to lab work. That to me, you didn't say too much about that, but I think anybody who does economic theory has to look at that and think of that as an incredibly, important piece of evidence and powerful indictment. Your thoughts?
Yeah, I think it's fair enough what you say. My response would be we try to avoid too much speculation. We obviously have to speculate a bit, but what we try and do is stick to what we actually know, or don't, and draw attention to that rather than to look for motives behind what's going on. Yes, I strayed into looking at motives a little bit in a few parts of my talk. And on that, it's just worth bearing in mind that the Xi Jinping regime would be just as angry if the market got blamed as the lab. That market belongs to some kind of cousin of his. And he has championed traditional Chinese medicine, which includes the use of pangolin scales as a medication at the World Health Organization and other places, etc. So you can see why, when the World Health Organization went over there and investigated, the Chinese scientists insisted that the most likely explanation was that that virus had been imported into Wuhan from abroad on frozen food, for which there is zero evidence. I mean, that really is as sort of useless a theory as you can come up with. The best they could come up with to absolve both the market and the lab. So there's a very strong Chinese motivation not to be blamed at all for this, and not to have to you know, to go through the sort of humiliation of the rest of the world looking into their affairs. So I think a lot of that, you know, a lot of the secrecy doesn't necessarily come from, doesn't have to come from guilt. It could come from just amor proprio, you know, keep out, this is our territory, etc. But imagine any other country in the world, Brazil, Russia, Britain, you know, anywhere you like, this kind of thing happening. The media and other organizations would not have let it rest till they found out what had happened. I think there is, you know, China is so powerful a country, so influential in the World Health Organization and others, that it is a bit different in that respect.
And the, you know, the other possible explanation you just gave, you know, keep out, we just don't want you searching, seems unlikely to me if you think of strategic actions because of the inference people will make from that. And, you know, the only sensible way to rationalize that's the actions is that to me, is that the Chinese government believes, maybe they're wrong, but believes they know something that will look very bad to them.
Yeah, I mean, in January 2020, a Major General in the People's Liberation Army was sent in to run the lab. That sounds pretty scary when you think about it, but she's actually a virologist. So, you know, it did make some sense, but you know, there's undoubtedly been a significant centralized control of information going on here. And yes, as you say, the easiest way to get people like me off their back is to publish that database.
How reluctant have been academics to look into these areas and discuss it in your?
Well, this is one of the really disappointing things, is that Alina and I get a lot of private encouragement from academics. People who say, keep going, you're on the right track, it's very important. But I don't want to put my head above the parapet. We hear that far too often. And a relatively small group of virologists are very, very active with the help of certain sort of science journalists in sort of pushing the case closed. We know it's the market. Can we shut up, please, and stop having the argument? And that you're seeing a lot of that just in the last couple of weeks because they're trying to head off a congressional hearing. Trying to say, look, you don't need to look at it. It's all closed. We know what's happened. I don't fully understand the motivation there. I'm very disappointed in scientists in taking this attitude. It feels to me like, yes, there will be consequences for the funding of virology, for biotechnology in general, for public approval of science. You know, those could be kind of that I wouldn't like either. I'm very pro-biotech, I'm pro-vax, you know, and all that kind of stuff. But for me, as I think Solzhenitsyn said, truth is more important than consequence. You know, we still have to find out the truth and then deal with whatever the consequences may be. I don't want to get into the business of accusing academics of having significant Chinese funding or connections, but that is true in some cases too. You know, I approached the Royal Society in London and said, President or ex-president, a great friend of mine, a Nobel Prize winner, and said, look, you know, we've got to have a debate. And he put me in touch with people, and they said, sorry, too controversial. I approached the Academy of Medical Sciences. They said the same. It's too controversial. What does that mean? How can it be too controversial? It's the whole point. The controversial subject. We should debate it.
There are a few questions online also. Jonathan?
Thank you, Matt, for a phenomenally interesting talk. And let me just apologize for not being in the room. The day change, I had to rejuggle my schedule to make the talk. I want to go back to your second point, because I think that to me is the really dangerous thing. I mean, look, as far as I'm concerned, it's not even a debatable case anymore. Jeff's argument is overwhelming. Every single piece of evidence points to this coming from a Chinese lab. So it's pretty obvious that that's where it came from. The problem I have is if you were going to design an experiment to see, well, what could we do to infect the world? This is the experiment you would design. And it worked spectacularly well. So if we accept that it came from a Chinese lab, if we accept that they engineered the virus to be more vigilant, and then we accept the result of that, isn't this, a frightening event in the history of human beings?
Yes, is the short answer, but I don't want to go as far as to say, therefore they had malicious intent.
No, I agree. I agree. It's unlikely that they would have had a malicious intent because Chinese people were people who died first. So no, I don't think that's what happened. I think it was a mistake. But the mistake was an experiment to show how effective this technology is in killing people.
Right. And, I consider myself someone who follows molecular biology quite closely. I had no idea these kinds of experiments were going on. And if I had known, I don't think I'd have been in favor. I mean, I hope I would. It just seems to me that the risk-reward ratio here is way out of whack. The risk is huge, the reward is tiny. And what's the motivation then for them to go on doing this? I think there is a tendency in science to do what you can do and to be driven by, what's the spectacular thing that will get me a paper in Nature? And in that process, you sometimes forget to look over your shoulder and think, should I really be doing this? So that's the best I can come up with.
Could I follow up on this point?
Yeah, John.
So Jonathan's right. It looks very well designed to be researched to make it more infectious to humans, but not more lethal. This was on the scale of pandemics, this was spectacularly unlethal.
Correct.
So I would mix incompetence with devious if you want to infer motivation, we're working on a program to make viruses that are very bad for people. And along the way, we made it more infectious to people, and then there was incompetence and escape from the
Yes, although one of the things the pandemic has taught us is that a virus doesn't have to be that lethal to disrupt the world spectacularly.
But that this was an intentional leak in order to disrupt the world doesn't make sense.
No, I agree.
But that this was a leak of incompetence in a program that may well have been intended to disrupt the world seems more likely.
Yes, but I see no direct evidence for that last point, that this program was intended to disrupt the world. The Wuhan Institute of Virology was a dual use lab. It did have PLA interests in that. But if you look at the Chinese government's approach to biological warfare research, it looks just as defensive as America's. It looks as if they're doing this work to stay ahead of terrorists and bad actors and North Koreans, just like we do, if you see what I mean. It doesn't look like it's designed to produce a weapon.
Isn't the ideal, I'm sorry,
No.
still fine. Isn't the ideal to have a virus that you also have the antidote to? Not necessarily. That's the ideal offensive weapon.
Yeah, and one of the Republican minority reports a few months ago raised the question of whether or not they were surprisingly quick at getting a vaccine out. And the experiments they described having done with this vaccine to test its efficacy involved animal experiments that would have taken 4 months, which means they would have started before the pandemic began. So there's a hint there that possibly in the autumn of 2019, they realized they had a slow burn problem beginning, and they began work on a vaccine before telling the world that it was a virus or the loose. I haven't even got to the question of how their reactions in January 2020, when they could have localized and stopped the pandemic and acted differently, instead of pretending that it was all under control and Chinese New Year could go ahead in Wuhan, etc., is irresponsible as well. Lee?
Hi, I'm Leo Hanyon. I'm an economist at UCLA. Thanks for the fascinating talk. I'd like to ask you if you're familiar with a virologist named Robert F. Gary. I'd come across a piece by him published in the Proceedings of the National Academy of Sciences. It was published last fall, The title of the paper, I'll put in the chat box, is SARS-CoV-2 Furin Cleavage Site Was Not Engineered. I'm not a virologist, so I can't assess the 5 arguments he makes against the engineering the engineering hypothesis. So I was curious if you were familiar either with him or this paper, and if you had any thoughts about that. I'll go ahead and put the citation in the chat box.
Thank you, Lee. Yes, I know Bob Gary well. He's one of the authors on that proximal origin paper. After his paper came out, he appeared on the Megyn Kelly show opposite my co-author, Alina Chan. Or actually, he wouldn't appear with her, they had to do it sequentially. But she argued against the 5 points he makes. I won't go into them here because I'll probably mess it up. But yes, it's a contribution, but we don't find any of his arguments particularly convincing. I mean, essentially, he's arguing that it came about by recombination, that out there's another virus with a furin cleavage site that came along, co-infected an animal, and a jumbled version of the virus came out with this thing in exactly the right place. It's possible, but it's a bit of a magical thinking explanation, frankly.
Thanks very much.
I just wanted to ask, how were you able to obtain some of those documents that you showed us were redacted fully, and then you were able to?
Well, I shouldn't take the credit. Various freedom of information requests from journalists, particularly on the Intercept and various other sort of slightly less respectable ends of the media. US Right to Know, which is an anti-biotech organization that I don't normally want to find myself in bed with, and others have been diligent in pursuing freedom of information requests. Some of the stuff around the February meeting, Republican staffers on the Help Committee got to see those emails, but weren't allowed to photocopy them, but were allowed to transcribe them, which is partly why there's a few typos in one of them.
Can I ask a related question?
Yes.
This is the lawyer in me. Why were they redacted? So the federal government has the freedom of information request. You're allowed to see things, and there is a list of reasons why. And it could relate to national security, and it can relate to like personal matters. So it's like, my address or things about my spouse or something like that. But I didn't see it. I mean, I don't have that exhaustive list.
I think if I remember right, the excuse was this could lead to personal persecution of the scientists involved. Something like that.
But that's true of everything.
Correct.
Okay. I mean, that's super revealing.
I don't know. I can't remember the reasons given at the time. Eddie Holmes's correspondence we would particularly like to see, because he was actually in Wuhan when it started, and he was very closely working with the Wuhan Institute of Virology. He's Australian. He's a Brit, but he's based in Australia. And the Australian authorities have refused to agree to any freedom of information requests. Likewise, Andrew Rambar in Edinburgh, we failed to see any of their correspondence. So it's ongoing, the attempt to find out more, but it feels like dragging this stuff out of people is worse than them coming forward. They actually now make the case, look, our emails about that meeting are now in the open. They show us discussing, as we should, the possibility and then rejecting it. What's wrong with that? And they have half a point there, that I've made it sound worse because we had to drag it out of them than it would have been if they had volunteered it. Question there.
So with the Chinese, the plausible deniability is kind of good enough. And I noticed, that there's also a lot of potential for motivated reasoning and complicity. I know that you're trying to give us the best evidence you can, but I was interested, you didn't say anything about the letter that was published by Daszak and others that seemed to really make a very hard statement and was put up in a way that was outside of peer review. And then also even the indication that these were the kinds of experiments that the US government said we shouldn't be doing. But then in fact, we were contributing to them in a way that was like, we wanted to keep our fingers in. So, you know, whatever you're willing to say about that, I don't even see the book, but that seems to be pretty significant and a reason why the US government doesn't want to go after this.
Yeah, well, the moratorium on gain of function experiments in the US between 2014 and 2017 is significant. It didn't come about because of coronavirus experiments, but coronaviruses were included. Ralph Barrick of University of North Carolina was significantly affected by that. He was about to do experiments, had to stop them, etc. There is a degree to which we kind of outsource them to Wuhan at this point. Don't think it's quite that explicit. I think Wuhan says we'll do them anyway. Whether or not sending funds through the EcoHealth Alliance from the American taxpayer during the moratorium to Wuhan to do experiments that were not allowed in the US is a breach of the rules, is something that Anthony Fauci and Rand Paul will no doubt continue to argue about and have done so already. That's slightly above my pay grade as a non-American, etc. And I'm more interested in what happened in China than the US. But if I can just, you also mentioned the Peter Daszak letter in the Lancet, and I didn't mention that, but the proximal origin paper that I did mention, the Christian Anderson paper, that convinced me that it wasn't a lab origin, was one of two similarly influential things. There was a letter in the Lancet published on the 28th of February 2020 with 27 authors, I think. It wasn't the 28th, it was 28 authors and 27th of February or something, something like that. The names have got muddled in my head. And the person who orchestrated that letter, as we later found out through Freedom of Information, was Peter Daszak. But he also said, I don't want it to appear that I've orchestrated this letter. And he declared no interests in that letter. He said that there are no interests to declare. It took us, I mean, I appeared before a parliamentary committee in the UK on this and was able to press the editor of the Lancet, who said as soon as we found out that he had conflicts of interest, we demanded that he do a conflict of interest statement. So I said, well, why did it take you 18 months to publish that conflict of interest statement? Well, it took us that long to persuade him we should do it. So this is not very convincing. So in that letter, Peter Daszak basically said, and I was hearing this at the time, it's very unfair the way people are beating up my lovely Chinese colleagues. We should all science come to their support, etc., etc. Fine to say that, but add, oh, and by the way, I go to karaoke bars with them. They're my closest friends. I give them a lot of money. I publish a lot of papers with them. I have a major conflict of interest, and and have a lot of motivated reasoning. But he didn't do that. And by the way, if you remember, the EcoHealth Alliance's funding was stopped at one point by the Trump administration once all this came out. Something like 150 Nobel Prize winners then wrote saying this was outrageous. It resumed. It continues to this day. Peter Daszak was in a bat cave collecting samples from bats and boasting about it a few months ago in Thailand, as it happens. It's quite extraordinary, really, that this goes on. So this, it was very interesting. Jonathan said something like it's a slam dunk, we know what happened. An awful lot of the public think that, but nothing has really changed in the official circles.
Bariba, Elaine?
Yes, hi. Thank you very much for the fabulous talk. Very informative. I have a question, or actually, I don't want to kind of like bring any doubt to the source that you're talking about, but add to it. If you have done any research to what is going on also in some of the biolabs that now we are becoming a little bit more aware of their existence in Ukraine. And if any of this stuff is happening, and especially with the war, what kind of danger the world could be in, because there are a lot of labs that, from what we understand and we've heard, that they have been funded in Ukraine, and it could be in hands of all different types of people, including terrorists. So how can we kind of like start with this experience, think about what we can do about preventing the world from falling into another situation similar or even worse. So I don't know if it's open to you're open to expanding to that discussion, or maybe at a later talk you can talk about.
I'm afraid I don't know enough to respond in any meaningful way about what's going on in Ukrainian labs. When the war began, there was a few remarks about some labs might be implicated in this whole story, but I couldn't see the link myself, so I haven't really followed up on it. So I'm sorry, I just don't know the answer, but I've not seen any evidence of anything like this happening in labs in Ukraine.
Okay, thank you.
Thank you very much, Mike.
Well, no, not quite. I'm meeting plans to by the San Francisco.
Yeah, let's do it. Yeah, we would. Do you want to join us? Oh, there is pulling it. Okay, so let's do that. And I think Jonathan also.
Just a interviewing going.
I read something. Yeah. Because we didn't know.
Yeah, well, I don't want to.
But it's the fact that he was.
Any of that.
Seated himself.
Quitaron la idea, ¿no?
I told him that he would be late.